Welcome students. Today our topic of discussion is seed, a unit of genetic recombination and replenishment. In spermatophytes, the seed is the final product of sexual reproduction. It's often described as a fertilized mature ovule that develops in the ovary of a flower. A seed typically consists of seed coats, one or two cotyledons or stored material, and a rudimentary plant called an embryo. The outer coat is called testa and the inner one tegmen. If there is only one covering, then it is called testa. The cotyledons of the embryo are simple structures, generally thick and swollen due to storage of food reserves. The embryo is surrounded by a nutritive tissue called endosperm that stores large quantities of carbohydrates, lipids, proteins and minerals, and that supports the growth and development of seedling until it establishes itself as a photosynthetically active plant. Mature seeds may be non-albuminous or albuminous. Non-albuminous seeds have no residual endosperm as it is completely consumed during embryo development. For example, in pea, groundnut, etc. Albuminous seeds retain a part of endosperm as it is not completely used up during embryo development. For example, in wheat, maize, barley, castor and sunflower. Normally, the nucellus, that is the central part of an ovule, is absorbed and is absent in seeds. Occasionally, it may persist in the seeds of some genera such as nutmeg, clove, papaya, pepper, beetroot, etc. as a thin layer called a perisperm lying inside the seed coat and supplies food material to the embryo. Integuments of the ovules harden as tough protective seed coats. The micropyle remains as a small pore in the seed coat. This facilitates entry of oxygen and water into the seed during germination. As the seed matures, its water content is reduced and seeds become relatively dry. The general metabolic activity of the embryo slows down. The embryo may enter a state of inactivity called dormancy, or if favorable conditions are available like adequate moisture, oxygen and suitable temperature, they germinate. Seed is the final product of sexual reproduction. Sexual reproduction is the production of offsprings by the fusion of two gametes to form a diploid zygote, which develops into the mature organism. Sexual reproduction allows the genetic information of two parents to recombine, that is genetic recombination, to form a new individual. Genetic recombination is a process by which a molecule of nucleic acid, either DNA or RNA, is broken and then joined to a different one. Recombination is a common method of DNA repair in living organisms, but in sexually reproducing organisms, it is also practiced by cells at the time of formation of gametes during meiosis. It involves two successive divisions, producing 4 germ cells with different chromosome combinations. Meiosis is an essential feature of life cycle in organisms where sexual reproduction occurs because it provides a mechanism for reducing the amount of genetic material by half. This ensures that when gametes fuse, the diploid number of chromosomes is restored. If the chromosome number were not halved before production of gametes, the chromosome number would double with every generation. One great advantage of sexual reproduction is the production of a great deal of genetic variation through the shuffling of both beneficial and deleterious mutations because of independent assortment of chromosomes and random fertilization. Random assortment of different alleles of genes on different chromosomes depends upon the segregation and independent assortment of the chromosome during meiosis 1. Independent assortment refers to the fact that bivalents line up independently and therefore the chromosomes in each bivalent separate, that is assort, independently of those in other bivalents during anaphase 1. Genetic variability is also produced by genetic recombination through the process of crossing over. Chromosomal crossing over refers to recombination between the paired chromosomes inherited from each of one's parents. When the chromosome pairs during meiotic prophase, parental homologous chromosome exchange segments during crossing over to produce recombinant chromosomes, leading to the formation of new combination of genes on the chromosomes of the gametes. During prophase 1, the 4 available chromatids are in tight formation with one another. While in this formation, homologous sites on 2 chromatids can mesh with one another and may exchange genetic information. Because recombination can occur with small probability at any location along chromosome, the frequency of recombination between 2 locations depends on their distance. Therefore, for genes sufficiently distant on the same chromosome, the amount of crossing over is high enough to destroy the correlation between alleles. Thus, the 4 cells, spores or gametes produced as a result of meiosis are all different. This produces offsprings with new combination of traits different from those combinations found in the parents, resulting from the events of meiosis and random fertilization. Secondly, the combination of 2 sets of chromosomes, one set from each parent in the zygote, also forms the basis of variation within species. These rearrangements alter the genetic constitution distributed among the parents. During seed formation, genetic recombination is performed by the plants at the time of formation of gametophytes after undergoing reduction cell cycle, that is meiosis, which facilitates crossing over. This gives rise to haploid pollen grains and haploid embryo sac. Thus, no 2 pollen grains are ever identical, nor are 2 embryo sacs. That is called DNA fingerprinting. These in turn form the male gametes and the egg, which are used for sexual reproduction to form a seed. It is clear that because of the genetic recombination, genome does not remain static and the cell's molecular machinery is altering the genome with each passing generation. Also, as a result of genetic recombination, the genomic constitution of each seed is unique in this world. Being products of sexual reproduction, seeds generate new genetic combinations leading to variations. These variations were observed by various scientists like Knight, Gauss and Mendel long before after carrying elegant experiments. Knight crossed a pea plant with green stem, white flowers and colorless seed coat with another one which had purple stem, purple flowers and brown seed coat. He obtained the seeds that yielded only colored plants in the first filial generation. On salving, these plants yielded pods with both colorless and colored seed coats. Similarly, while working with the laws of inheritance, Mendel crossed a round and yellow seeded plant with a wrinkled and green seeded one and observed that although first filial generation yielded plants with parental characters only, the second filial generations have characters different from that of their parents, as he was able to get the plants with round green and wrinkled yellow characters also, beside the ones with parental characters. This led to the formulation of law of independent assortment of genes and characters. Since seed formation involves independent assortment of genes and characters and crossing over at the time of formation of microspores and megaspores and then random fertilization, the seeds are rightly regarded as a unit of genetic recombination. Now, development of male gametes, female gametes and seeds in angiosperms. The 2 most important reproductive units in seed plants are stamens and carpels, producing male and female gametes respectively. They develop in a flower. The two parts of a typical stamen are the long and slender stalk called the filament and the terminal, generally bilobed structure called the anther. When the anther is young, a group of compactly arranged homogeneous cells called the sporogenous tissue occupies the center. Each cell of sporogenous tissue is a potential pollen or microspore pollen cell, that's PMC, that forms microspores through meiosis. The process is called microsporogenesis. As the anthers mature and dehydrate, the microspores dissociate from each other and develop into pollen grains. When the pollen grain is mature, it contains two cells, the vegetative cell and the generative cell. In most of the angiosperms, like 60% of the angiosperms, pollen grains are shed at this two cell stage. Now the carpels. The carpels present the female reproductive part of the flower. It has three parts, the stigma, style and the ovary. The stigma serves as a landing platform for pollen grains. The style is the elongated slender part beneath the stigma. The basal bulged part of the carpel is the ovary. Inside the ovary is the ovarian cavity, that's locule, where the placenta is present. Arising from the placenta are the megasporangia, commonly called ovules. An ovule consists of the nucellus, which contains the embryo sac surrounded by the integuments. Ovules generally differentiate a single megaspore mother cell, that's WMC, in the micropylar region of the nucellus. The megaspore mother cell undergoes meiotic division and produces 4 megaspores. In a majority of flowering plants, one of the megaspores is functional, while the other 3 degenerate. Only the functional megaspore develops into female gametophyte, that's embryo sac. Development of functional megaspore from megaspore mother cell is called megasporogenesis. The nucleus within the functional megaspore undergoes 3 successive divisions to form 8 nuclei. These 8 nuclei are arranged as 3 antipodal cells at the calyzal end, 2 central polar nuclei, 1 egg cell with 2 synergids at the micropylar end. The synergids have special cellular thickenings at the micropylar tip called filiform apparatus, which play an important role in guiding the pollen tubes into the synergid. Once a pollen grain lands on the stigma of a compatible species, it germinates, resulting in the emergence of a pollen tube from one of the pores of its walls. The contents of the pollen grain migrates into the pollen tube. Pollen tube grows rapidly through the tissues of the stigma and style and reaches the ovary. After entering one of the synergids, the pollen tube releases the 2 male gametes into the cytoplasm of the synergid. One of the male gametes moves towards the egg cell and fuse with its nucleus, thus completing the syngamy. This results in the formation of a diploid cell, the zygote. The other male gamete moves towards the 2 polar nuclei located in the central cell and fuses with them to form a triploid primary endosperm nucleus. The central cell after triple fusion becomes the primary endosperm cell and develops into the endosperm, while the zygote develops into an embryo that matures into a seed. Now seed as a unit of replenishment. The seed is not only an organ of perpetuation and dispersal, but also the major plant tissue harvested by mankind. The main food materials present in seeds are carbohydrates, proteins and lipids. These compounds occur together, but their ratio is different in different types of seeds. On dry weight basis, seeds of cereals contain 70 to 80% of starch. Those of peas and beans about 50%. Similarly, the amount of protein present in seeds varies from 10% in cereals to 40% in certain legumes and oil seeds of the dry weight, forming a major source of dietary protein. The seeds are also a rich source of oils. For example, the seeds of rape mustard yields 40% of oil and those of soybean 20%. These components make the seeds a unit of replenishment not only for animals but for the plants itself also. Resumption of active growth in the dormant embryo and development of young seedling are dependent upon the stored food reserves in the seed. The quiescent dry seed rapidly resumes metabolic activity upon imbibition. The structures and enzymes necessary for this initial resumption of metabolic activity are generally assumed to be present within the dry seed. The food reserves like starch, hemicellulose, phosphates, proteins, lipids are broken down enzymatically and mobilized towards the developing seedling. Seeds as a source of carbohydrates and their mobilization. Carbohydrates are the main storage reserves of most seeds. Beans, lentils, peas, whole grains, brown rice, buckwheat, millet, quinoa, yeast-free sprouted grain breads, nuts and seeds like walnuts, almonds, sunflower seeds, sesame seeds, and pumpkin seeds are good source of carbohydrates. These crops are widely used by human population throughout the world. The most common carbohydrate found in seeds is starch, though hemicellulose, amyloids, and raffinose oligosaccharides may sometimes be the major carbohydrate reserves. They also contain a great supply of plant proteins, fibers, and many other nutrients. During germination of seed, starch grains containing both amylose and amylopectin become corroded on the surface as hydrolysis sets in. Starch is normally broken down by alpha and beta amylase and a mixture of maltose and glucose is formed. In cereals, the breakdown of endosperm reserves depends upon the enzyme secreted by the aleurone layer. The biochemical specialization of the outermost endosperm layer as an aleurone layer is not restricted to cereals only. As observed in several members of Fabaceae, carbohydrate breakdown in the endosperm cells is concurrent with digestion of protein bodies and corrosion of walls in the endosperm layer. This layer thus appears to function as an aleurone layer in mobilizing its own protein reserves for synthesis of enzymes and excreting the latter through the corroded cell wall. The glucose resulting from hydrolysis of starch in cereal endosperm is taken up by the scutellum, synthesized into polysaccharides and transported to the growing seedling. The energy required in these processes is provided in the form of ATP by the actively multiplying mitochondria. In some seeds, carbohydrate reserves appear as thickened walls composed mostly of hemicelluloses, giving mannose and other monosaccharides. This type of storage occurs in endosperm of asparagus, iris, phoenix, strychnos, or cotyledons of impatiens, lupinus, primula, and tropolium. Now, seeds as a source of proteins and their mobilization. Seeds of legumes are the second most important source of food after cereals. Beans and legumes are inexpensive and a common food all over the world. Besides proteins, they are typically high in fiber, calcium, and iron. Legumes have globulins as a major storage protein, and unlike cereal proteins, they are rich in lysine content. Combined with whole grains like buckwheat, brown rice, and millet, beans and legumes not only make a delicious meal but often also provide the full complement of essential amino acids needed by humans. Seed proteins can be classified into 4 groups on the basis of their solubility. First, albumins. These are proteins soluble in water and salt solution. Second, globulins. These are proteins soluble in salt solution but insoluble in water. Third, glutenin proteins. These are proteins soluble in dilute acids or alkali solutions. And fourth, prolamines. These are proteins soluble in 70 to 80% aqueous alcohol. Many proteins, especially albumins, are enzymes and are metabolically active. They are widely distributed in dicot seeds and have been widely studied in the members of cruciferae, notably oilseed rapes, in which they are called napins. Although the vast of the individual proteins present in mature seeds have either metabolic or structural roles, all seeds also contain one or more groups of proteins that are present in high amounts and that serve to provide a store of amino acids for use during germination and seedling growth. These are called storage proteins. Storage proteins of seeds show much variation depending upon the species. For example, the major storage protein in maize, barley and sorghum is of prolamine type. In wheat it is glutenin type, and in oats globulins are dominant. The globulins are the most widely distributed group of storage proteins. They are present not only in dicots but also in monocots, including cereals and palms, and also in fern spores. These storage proteins are of particular importance because they determine not only the total protein content of the seed but also its quality for various uses. For example, the low content of lysine, threonine and tryptophan in various cereal seeds and of cysteine and methionine in legume seeds is due to the low proportions of these amino acids in the major storage proteins and may limit the nutritional quality of the seeds for monogastric animals. In the case of wheat, the storage proteins form the gluten fraction, whose properties are largely responsible for the ability to use wheat flour to make bread, other baked goods and pasta. These properties are not shared by the storage proteins of other cereals. Storage proteins are synthesized during seed development together with the reserve carbohydrates and lipids. Storage proteins must be distinguished from metabolic proteins that are concerned with normal cellular activities, including the synthesis of storage proteins. Storage proteins occur as diet reserve proteins which accumulate in vacuoles, and when the process is completed, the accumulation appear as bodies enveloped by unit membrane, presumably the original tonoplast. During germination, the protein becomes dissolved in situ and the protein bodies are replaced by vacuoles which may fuse into one large vacuole. Reserve proteins are degraded to amino acids and small peptides by proteinases and peptidases. The sequence of breakdown of proteins in some monocotyledonous seeds like cereals differs from that in seeds of dicotyledonous plant. In the former case, the major reserve proteins are present in aileron layer and the endosperm. Only a small amount of storage protein is present in the scutellum and the axis. However, it is this small fraction that may be hydrolyzed to provide amino acids for the growing seedling before to the mobilization of the major endosperm reserves. The hydrolytic activity of dicotyledonous seeds involves two steps. Number 1, there occurs modification of large storage proteins to the forms that are prone to hydrolysis, and second, degradation of the proteins into amino acids and small peptides. The major transported forms of amino acids from storage organs into the growing seedlings are amides, that's asparagine and glutamine. This is supported by the fact that in the cotyledons of various legumes, there is an increased activity of asparagine synthetase and glutamine synthetase. These are the enzymes involved in the synthesis of asparagine and glutamine. Higher in Pisum sativum, the major transported form of the amino acid is homoserine. In castor bean seeds, where the site of protein storage is the endosperm, some of the amino acids released from storage proteins by hydrolysis are converted to sucrose and transported then to the growing seedling. Now, seeds as a source of lipids and their mobilization. Lipids appear in the form of oil bodies in the cytoplasm of oil storing tissues, as for example in cotyledons of white mustard or in scutula of cereals. Lipid bodies are also referred as lipid containing vacuoles or spherosomes and are then assumed to have a bounding unit membrane or half of a unit membrane. Lipids are hydrolyzed in situ by lipases to yield glycerol and free fatty acids. Usually, none of the breakdown products of hydrolysis of lipids accumulate in seeds. The fatty acids are further mobilized chiefly by beta oxidation. The fatty acids are utilized for synthesis of phospholipids and glycolipids, which are required as constituents of organelles, but most are converted to sugars and transported to seedling body for growth. The glycerol becomes part of the general carbohydrate pool present in the seed and as such becomes available for various processes including respiration. The beta oxidation activity is associated with the glyoxysomes. The activity of glyoxylate cycle enzymes during germination shows that these enzymes become active on second or third day after imbibition when the radicle is about 2 millimeters long, and complete digestion takes a further 4 to 5 days. The liquefied contents are absorbed by expanding cotyledons attached to the growing root shoot axis. Thank you. With this I take your leave and hope to see you again with a new topic. Till then, goodbye.